Hi everyone. In this lecture, we will learn filamentous DNA bacteriophages. What are filamentous bacteriophages? Three filamentous bacteriophages that infect E. coli are M13 bacteriophage, FD bacteriophage, and F1 bacteriophage. Each of these filamentous bacteriophages contain a circular single-stranded DNA molecule, and they have been carefully studied. So like 5X174 bacteriophage, which we have seen in the previous lecture, these bacteriophages also contain single-stranded DNA as the genetic material, and it is circular. Let's see more about these bacteriophages. Why are they called as filamentous bacteriophages? If we see the structure of these bacteriophages, it is very unusual if you compare them with P bacteriophages or lambda phage. They have a filamentous morphology. There is a P3 protein and P6 protein at one side or one end of M13 bacteriophage, which is representative of filamentous bacteriophage. And there is P7 and P9 at the other end. The major coat protein is P8, which coats the DNA. P8 has positively charged lysine residues that attach to the negatively charged DNA. This is another picture of the structure of M13 bacteriophage. The P3 protein at the one end is the attachment protein, and that helps the bacteriophage to attach to the F pilus to enter the host cell. So if P3 is the attachment protein that helps the bacteriophage to attach to the host cell, the receptor which is used in attachment is the F pilus, which is also called a sex pilus. So which means that this M13 bacteriophage can only infect cells which have the F pilus. So the male cells, E. coli cells, which carry the F episome or the F plasmid. There is protein P6, as we have already discussed, P7 and P9 at the other end. The circular single-stranded DNA is supercoiled inside this bacteriophage. There are 5 promoters and 3 terminators. This is the genome or the genetic map of this bacteriophage. Of all the known virulent bacteriophages, these filamentous bacteriophages do not kill the cells. So of all the known virulent bacteriophages, these filamentous E. coli phages do not kill the host cell, nor do they cause lysis. So how are they gonna released if they do not kill their host cell or do not cause lysis? They convert the host cell into a phage producing factory, which is in the case of other bacteriophages as well. But after that, they do not kill the host cell or they do not cause the cell lysis. They are released via budding. They may reduce the growth of the or growth rate of the host cell, but they do not kill them. I've already mentioned that these filamentous phages adsorb to the tip of the F pilus, and the particle is brought to the cell wall at the base of the F pilus. The pilus retracts by the absorption of the pilus proteins into the bacterium. We are going to see these 3 points with the help of 2 diagrams. So in the first diagram, a comparison is shown between, let's say, a T bacteriophage and the filamentous phage. On the left, what is visible on the slide is a T bacteriophage, and how these bacteriophages have an icosahedral head, a collar, helical sheath, there is core inside, hexagonal base plate, and the tail fibers. When the tail fibers attach to the receptor sites and when the landing occurs where the hexagonal base plate sits onto the receptor sites or the cell surface. The tube is inserted and the DNA is injected into the host cell like a syringe. In contrast, the filamentous bacteriophages, which infect male or F plus E. coli cells, and the receptor site is the F pilus, the P3 protein attaches to the F pilus, and then the pilus retracts. Pilus retracts or contracts, and the phage is brought to the cell surface. Let's see a detailed diagram which shows the attachment of the P3 protein of filamentous bacteriophage such as M13 to the receptor site, which is the F pilus. This is a M13 bacteriophage, and this is the coat protein P8, which is coating the genetic material single-stranded DNA circular. This is the P3 protein which is involved in the attachment. And the P3 protein here attaches to the F pilus or the sex pilus. And after the attachment, the pilus contracts. In the next step, the pilus has contracted. And once the pilus contracts, the phage is brought near to the cell membrane, where the P3 also interacts with another receptor present in the cell membrane of the E. coli cells or the host cells, that is tall A. So can I say here that the attachment protein or the ligand P3 is attaching to the F pilus as well as tall A? Yes. F pilus is the main receptor and tall A is the co-receptor. We have seen the involvement of more than one receptor and the main receptor and the co-receptor in case of animal viruses such as HIV, where how the GP120 protein of HIV interacts with the CD4 as the main receptor and then CXCR4 or CCR5 as co-receptors, depending on whether it's a CD4 positive T lymphocyte or a macrophage. So here it's a bacteriophage that is using a co-receptor for the attachment. The main receptor is the pilus and the co-receptor is tall A. What happens next? The entire particle in intact form penetrates the cell wall and comes to the rest of the inner cell membrane. So we can contrast, have a different, we can talk about differences between these filamentous bacteriophages and let's say T phages. So how these phages do not kill the cell or do not cause cell lysis in contrast to T phages and other lytic phages. One more difference is that the entire particle in the intact form penetrates the cell wall and comes to rest on the inner cell membrane. The particle is uncoated in the inner cell membrane and the single-stranded circular DNA is released, which is converted to a double-stranded form. We are going to see this step in detail in the next slide, where we'll talk about the replication of this bacteriophage. The coat is degraded to release the single-stranded circular DNA genome, and these protein subunits are reused or recycled when the progeniphages or progeniphage particles will be assembled. The entry of the coat proteins into the cell is unique to these phages. One more difference. Like a T bacteriophage, a T4 bacteriophage will only inject its DNA and the rest of the proteins will remain outside the cell surface. Here, coat proteins also enter into the cell and these are unique to these bacteriophages. Let's see the replication of M13 bacteriophage. After the attachment, once the genome is released into the cytoplasm, this genome is DNA, single-stranded DNA, circular, as we can see in this diagram. It's a plus strand. Transcription cannot occur for this circular single-stranded DNA. So this single-stranded DNA has to be converted into a double-stranded DNA form. And this single-stranded DNA is converted to a double-stranded DNA with the help of E. coli or host enzymes. And this double-stranded DNA, once it is made, it's called as replicative form or rfDNA. It can also be called as RF1, replicative form 1. After this double-stranded DNA is made, via the various promoters, transcription occurs, resulting in the mRNA molecules, and the RNA processing also takes place, followed by translation. Once the proteins are made, so far what we have seen, although I'm calling this as replication, this is replication and gene expression. So far we have seen the transcription and translation. Once the proteins are made, the replication will begin, and like 5X174, these bacteriophages also replicate via rolling circle replication, which is the case for many viruses. And there are many similarities to 5X174 replication here. A nick will be made, the strand would be displaced, and the rolling circle replication would take place, which will result in a single-stranded circular DNA displaced plus strand. So the replication is happening or it is over. What happens next once this single-stranded circular plus strand is made? The next step, whether this single-stranded DNA will go for the formation of double-stranded DNA, that is replicative form again, or this single-stranded DNA would be packaged and the assembly process would begin. This depends on the concentration of a protein called as P5 protein. So, how is this P5 protein significant? If the concentration of P5 is low, if P5 is in low concentration because enough transcription and translation has not happened yet, then under low P5 concentration, this displaced strand will go for the synthesis of double-stranded DNA, which is replicative form 1. This is going to happen in the low P5 concentrations, and once this replicative form is made, it will again go for transcription and generation of mRNA, which will be processed, and more protein synthesis. But a time would come when the concentration of P5 inside the host cell would increase. When there is high P5 concentration, the P5 protein is going to coat this single-stranded circular DNA, this one, and will form a pre-assembly ssDNA complex, which means the P5 after coating or by coating this displaced plus strand, single-stranded DNA circular, doesn't allow it to go for the synthesis of the replicative form or the double-stranded DNA. So once this pre-assembly ssDNA complex takes place, this will move towards the cell membrane for assembly. The major coat protein, what we have spoken about in the initial part of the lecture, gene 8 protein or P8, which coats the DNA, it is synthesized in large quantities because it's required in large quantities, and it's immediately deposited in the inner cell membrane. Some references also say that the assembly process is poorly understood for M13 because unlike other bacteriophages, the assembly takes place in the inner cell membrane rather than cytoplasm. No gene 8 protein or P8 is found free in the cytoplasm. It is found only in the inner cell membrane, and it's a alpha helix protein having 3 distinct regions, a basic end, a hydrophobic center, and an acidic end. The hydrophobic center of the protein enables the proteins to be situated in the cell membrane, which is hydrophobic. The acidic end of the protein faces the cell wall outside, and the basic end of the protein faces the cell interior. And once the pre-assembly complex approaches this P8, perhaps this P8 will displace the P5 or the gene 5 protein, which is coating the DNA, and this P8 will coat the plus strand, and assembly has begun. The phage particle is assembled in the inner membrane, and the other proteins P3 and P6 on one end and P7 and P9 would be attached. For each completed particle, a bud forms on the cell surface and the phage is extruded. This process can continue indefinitely without damage to the cell. As we have said in this recording, that these filamentous bacteriophages, M13 bacteriophage, they do not kill the cell, they do not cause cell lysis. So the phages can be produced indefinitely without doing damage to the cell. In a way, when the cell doesn't get damaged, it continues to keep on making more phages. So it works to the advantage of the bacteriophages. And these phages are, if they do not cause cell lysis, how are they released? They are released via budding. So in this lecture, we have learned what are filamentous bacteriophages, their examples. We have learned M13 in detail, its structure, attachment, entry, gene expression, replication, and assembly. And we have also compared how these bacteriophages, filamentous bacteriophages such as M13, different from other lytic phages.